Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Professor Emeritus in Department of Biochemical Engineering and Biotechnology at IIT Delhi. Today we are going to talk on the module which deals with complement system. We will discuss this complement system in couple of modules. So in that way, this is the first module which deals with complement system, and this is of course from the paper on immunology. What we call complement system is actually a set of serum enzymes. That's where the complement system is present in the serum, blood serum. Complement system is also a very important illustration of how cascade systems are used in biology, in biological designs, to amplify a biological signal. You would have come across similar cascades, or you will come across similar cascades when you look at the metabolic controls, even in the common metabolic pathways like glycolysis or glycogen degradation. Perhaps the most familiar example of the use of the cascade is the simple blood clot formation. The objectives of this particular module are then to understand complement as a cascade system of reactions that obviously will also indicate why the use of a cascade system is a good way of amplifying signals in this context as well. We will also learn about how to measure and detect complement. In many contexts, and largely in the medical context, the diagnostic context, detecting the complement and measuring it is actually very important, and hence rather than we will talk more about it when we talk of the toolbox on the immunology, but this is a good place where we actually cover this simple techniques of measuring the complement and detecting it. In this module, we will explain how classical pathway of the complement works. There are three important pathways of the complement activation. Classical pathway is one of them. We will also understand how an alternative pathway, which is the second pathway for complement activation, this operates. It is important to understand and remember that unlike the classical pathway, the alternative pathway is not dependent upon humoral immunity. So the concepts involved in this module are that the major functions of the complement system are initiating inflammation, attraction of neutrophils, and opsonization of the microbe, and finally killing of the microbe by lysis. The complement system is common to all vertebrates. It is one of the serum enzyme systems. So, in this design, cascade design, is one reaction happens initially, and this leads to form the active enzyme for the next reaction, and this goes on down the line. This results in a domino effect where a small stimulus can get amplified down the line. In humans, complement contains 20 soluble glycoproteins. Many of these are produced by hepatocytes and monocytes. These are constitutively present in blood and are designed as C1, C2, etc., or as factor B, C, etc. In many cases, uncontrolled activity can have disastrous consequences, yet when needed, it needs to be in place when required. The strategy is to limit such activities by producing them in small quantities and with short half-lives so that the high amount of activity is generated transiently. Cascade system like complement achieve this beautifully. Complement system was discovered by Belgian scientist Bordet. It is a system whose activation results in disruption of cell membrane. There are various pathways by which complement system operates. In the classical pathway, the trigger is an immune complex of antigen and antibody. The properties of antibody molecules complex with antigen molecules are very different from free antibody molecules. Firstly, the network of antigen and antibody molecules create a spatial array of molecules. Secondly, antigen binding to antibody via Fb fragment generates a conformational change which leads to Fc region being more available for many interactions. The other consequences of this antigen-antibody complex formations are listed here and include chemotaxis and phagocytosis, degranulation of mast cells and basophils, initiation of the complement cascade, which we are discussing now, platelet aggregation and degranulation, generation of K cell activity, lymphocyte division and differentiation. This slide shows classical complement pathway. Complement factor 1 can be stimulated by IgM bound to antigen or pair of immunoglobulin G bound to the antigen but located closely on the antigen surface. Although the complement components are numbered, the sequence of their participation is not in numerical order. Thus, C1 acts on C4 and so on. Polymeric IgM activates complement more effectively than immunoglobulin G. This table here shows components of all the pathways of the complement activation. While right now we are discussing the classical pathway, other pathways have some overlapping components and some different components. This table introduces all the components for all the pathways. We have already seen the beginning of the classical pathway. Out of this list, C1, C4, C2, C3 are involved. Let us look at their involvement and the rest of the classical pathway. One of the important intermediates of the classical pathway is a macromolecular complex. These 5 components in this complex, C1Q, 2 molecules of C1R and 2 molecules of C1S, are bound to each other non-covalently. This non-covalent association is stabilized by calcium ion. C1Q itself consists of 18 polypeptide chains, in which 3 polypeptides A, B, C form a subunit. C1 is made up of 1 molecule of C1Q and 2 molecules of C1R and C1S respectively. The chains are linked by disulfide bonds. 80 residues at the N terminal have a collagen-like structure, with glycine occupying 3rd position in a sequence rich in hydroxyproline, hydroxylysine, and of course glycine. In fact, collagenase can digest part of C1Q. The complex, which consists of dimers of C1R and C1S, can occur as such and has an extended form. Upon binding with C1Q, it turns into a more compact form twisted with shape of 8 as shown in this slide. Each C1R and C1S has one catalytic domain and one interaction domain, which is responsible for binding with each other as well as with C1Q. Binding of C1Q brings catalytic domains of C1Q and C1S together. The globular heads of C1Q, the C terminals which do not have collagen-like structure, that is responsible for binding to the Fc portion of the antibody, which in turn is already bound to an antigen. Each C1Q molecule has to bind to at least 2 Fc sites for a stable complex formation to take place. This is the molecular mechanism of the trigger, which requires either 2 IgG molecules closely or an IgM bound to an antigen. Also, in circulating IgM, C1Q binding sites on Fc are not exposed. Hence, unless bound to an antigen, IgM cannot initiate complement cascade reaction. This acts like as a safeguard for unwanted triggering of the complement cascade. C4 is the next component which comes in play. It is a glycoprotein with 3 chains alpha, beta and gamma. Their molecular weights are 93 kilodalton, 78 kilodalton and 33 kilodalton respectively. C1R and C1S have potential catalytic domains. Coming together of these domains results in C1R getting activated and splitting C1S. This exposes catalytic sites on C1S. Consequently, C1S cleaves C4. The active C1s in fact cleaves alpha chain of C4 to form 2 fragments. A small fragment from the N terminal called C4A and a major fragment called C4B. Let us look at the structure of C4. C4B is unstable in free form and can be rapidly broken down to an inactive product. C4B can however bind to neutrophils and platelets and lymphocytes, as all these cells have the complement receptors on their surface. C4B can also continue to participate further in the complement pathway. Activated C1S can also split C2. C2 is a glycoprotein of 117 kilodalton. C2A can combine with C4B. C4B C2A is called the classical C3 convertase as it activates C3. C4b C2a can further complex with C3b to form C4b C2a C3b, which is called C5 convertase. This makes C3 as a very important component of the complement system. C3 has molecular weight of 190 kilodalton and has one alpha chain which is of 120 kilodalton and one beta chain which is of 70 kilodalton. Both these chains are linked by two disulfide bonds. C3 has the highest concentration among all complement components in the serum. In a way, C3 activation is the most critical step of the complement action. C3 can also be activated by other pathways. In fact, all pathways differ in this early stage and the characteristic way in which C3 is activated. The late stage of all the pathways is in fact similar, in which essentially large multimolecular complexes of other complement components cause cell lysis by damaging membranes. Let us now look at the beginning of the alternative pathway. C3 actually can also break down spontaneously in plasma, and as a result C3b is generated in normal serum at small concentrations all the time. Because of this, C3b can combine with another protein to form alternative pathway which is based upon C3 convertase. Thus, through this route, C3b can form an adequate amount. A protein called factor H which can destroy C3b very fast. For alternate pathways to operate, hence factor H activity has to be controlled. Factor H is a glycoprotein that can bind to alpha chain of C3b. This makes alpha chain of C3b susceptible to proteolytic attack by factor I. Factor H requires surface on the antigen which is rich in sialic acid to be able to bind C3b. For example, if some bacterial surface do not have enough sialic acid, factor H cannot bind to C3b. In that case, C3b is able to bind to factor B. This leads to progressive activation of the complement pathway by positive feedback loop. We introduced a new factor called factor B. Factor B is a large protein of 100 kilodalton molecular weight. Its binding to C3b is magnesium dependent. C3bb has weak C3 convertase activity. It can, however, acquire potent C3 convertase activity upon cleavage by a factor called factor D. C3bb is a potent C3 convertase. Factor D is a serine protease which actually cleaves B only when it is bound to C3b. So C3b regulates this process. C3bb has a very short half-life of 5 minutes. Its half-life is extended to 30 minutes if it binds to another protein called factor P, or which is also called properdine. Factor P is a gamma globulin of 220 kilodalton molecular weight and can bind to C3bb reversibly. Because of the involvement of properdine in a critical fashion, sometime alternative complement pathway has been referred to as properdine pathway. This slide shows the two possible alternative fates of C3b molecule. The alternative pathway requires low level breakdown of C3. This cleavage generates a thioester bond in C3b, which is reactive enough to bind to many molecules or cells. If C3b binds to a cell, there are regulating molecules which can inactivate it. If it binds to the invading microbe instead, Bb binds to this bound C3b. The alternative complement pathway can protect from infection unaided by lymphocytes. Actually, alternative complement pathway is older pathway in terms of evolution. It may be recollected that antibody formation takes some time to form. Hence, during this period, alternative pathway is the first line of defense. Classical pathway in fact evolved much later after the evolution of humoral immunity. Let us summarize what we have learned so far. So, what you would have seen is that it is at the C3b stage, formation of the C3b molecule, that both classical pathway and the alternate pathway converge. From this point onwards, it is called the terminal part of the complement pathway, and this terminal pathway is common for both the classical pathway as well as the alternative pathway. So, C3b formed from either classical pathway or alternative pathway initiate the late phase or terminal complement pathway. C5 convertase is formed from either of the pathway by binding of the components. C5 convertase in the classical pathway is a complex of C4b, 2a and 3b. On the other hand, C5 convertase of the alternative pathway is PC3b2bb. C5 is a protein of molecular weight of 206 kilodalton with an alpha chain which has a molecular weight of about 141 kilodalton and a beta chain with a molecular weight of 83 kilodalton. Both alpha chain and beta chain are linked by a disulfide bridge. C6 is a single chain protein of 95 kilodalton molecular weight. C7 is a protein of 120 kilodalton molecular weight which can bind to C5b C6 complex either in free solution or when the latter is bound to a membrane. Membrane bound C5b C6 C7 binds to C8. C8 is a 3-chain protein of molecular weight 163 kilodalton. Formation of C5b C6 C7 C8 initiates polymerization of C9. C9 is also called perforin. Its oligomers of 12 to 15 monomers form a poly C9 which can damage the integrity of the cell membrane by insertion. The central role of poly C9 becomes the formation of a transmembrane channel resulting in osmotic lysis of the cell. The alternate pathway was discovered by Pilmer in the 1950s. So, it was actually discovered after classical pathways. Unlike classical pathway, it does not require antibody bound to the microbe. So, what actually triggers it? And what can trigger it? It can get triggered by presence of many substances. We will also see that apart from these two, there is a yet another pathway which can also activate the complement. It should be made clear that the alternate pathway can be viewed as a part of the innate immunity, whereas classical pathway, since it is dependent upon antibody, is a part of the adaptive immunity. So, what decides which pathway will be triggered? The alternative pathway, which as we mentioned earlier, was the one which evolved first and is actually not dependent upon the adaptive immunity or its humoral component. The alternative pathway can be triggered by microbial polysaccharides like inulin. It can be triggered by lipopolysaccharides of a gram-negative bacteria. In the case of infection by a gram-negative bacteria, it can be triggered by the teichoic acids which are present on the surface of the gram-negative bacteria. It can also be triggered by bacteria and parasites such as Schistosoma mansoni. So, encounter with a foreign surface can trigger it. Deficiency of complement components is associated with many diseases. Let us look at the assay for the total hemolytic complement or determination of a parameter which is called CH50. In this assay, antibody coated RBC are added to the test sample of the serum. Normally, sheep red blood cells are used. With this, the classical pathway is activated and antibody coated sheep RBCs are lysed. The sera as a source of hemolytic complement vary in their efficiency. Guinea pig complement is highly hemolytic, whereas horse complement is non-hemolytic. Hence, generally guinea pig complement is used here. 1 CH50 unit is the amount of complement required to lyse 50% of a standard preparation of sheep RBCs. In the presence of calcium cations, chelators like EDTA, C1 is inactivated and classical pathway is blocked. If necessary with such samples, the alternative pathway is activated by adding an endotoxin or immunoglobulin aggregates. The above test is based upon the non-specific nature of complement. Complement from one species can work with antibodies and antigens from another species. Complement content does not change upon immunization. All immunization does is to induce the activation of the different pathways. The lytic property of complement is thermolabile. Heating at 56 degrees centigrade for few minutes destroy its lytic activity. This is useful property. To rule out the artifact in determining CH50, the serum sample can be heated and the test redone to obtain a control zero value in the CH50 determination. The individual complement compound can be quantified using immunoassays. As deficiency of each complement component is known to be responsible for specific diseases, this serves as a useful diagnostic test. The complement in the test serum of the patient can be used as a chemoattractant and measuring neutrophil chemotaxis. This evaluates chemotactic factors such as C5 alpha. In fact, this attraction of neutrophils to C5 alpha leads them to the site of attraction and constitutes one of the major functions of the complement. So, let us summarize. Complement can destroy a microbe with or without the help of lymphocytes. In the earlier evolved pathway, which is the alternative pathway, that is activated by the presence of foreign surfaces, the classical pathway is triggered by antibody coating a microbe. Various assays for detecting lytic activity of the complement or estimating its individual components have been developed. The complement system is a powerful weapon as a part of our immune defense system. It has many biological consequences. It also needs to be regulated. In this lecture, we have focused on its activation and lytic property only. Thank you.